Oh,
I think we're live. Yeah. Hey guys, welcome to the FARA Facebook Live series on the Grant Program. I'm Angel Martin.
And I'm Kyle Bryant.
Yeah, and this is actually our last episode of this series, and we're really excited to connect with you guys to talk about clinical infrastructure.
So as you know, this whole series of videos is about the FARA Grant Program.
Mm-hmm.
And for the first part of the series, we talked about 3 episodes. We talked about how FARA supports research in the lab to advance approaches towards the clinic. And so this episode, we want to talk about how FARA helps develop the clinical infrastructure to ensure success in the clinic.
Yeah, exactly. And when we were talking with the crew here about, just the importance of this category, so you'll see on the curefa.org/grant website, right, that just like with drug discovery, we're investing quite a bit every year into that category. We're investing, quite a bit as well into the clinical infrastructure categories, which we'll talk about in a minute. And, kind of historically, the reason why is because, I think we understood early on that, say that we do develop, right, a drug, or a drug is ready, right, to be tested in an FA population, we have no actual infrastructure in place, right, to, make that happen.
Now what?
Yeah, right? We don't have the physicians in place at sites, right, or even the patients, ready to be able to participate in such a trial. And so the clinical infrastructure, what we mean by that, right, is going to be our patient registry, our, clinical or collaborative clinical research network, or in the CCRN sites, things like that. So, yeah.
So what you're saying basically is that we develop clinical infrastructure so that we're trial ready.
Exactly.
When it comes time to do a trial, we have the ability to do that.
Yes. Yeah.
And one other thing that I was thinking about, a benefit of clinical infrastructure is clinical research, like understanding more intimately the details of how FA affects somebody and somebody's life and their body. And I think that allows us to focus on, to kind of go full circle and have a more accurate approach to another possible treatment from the beginning.
It made perfect sense. Yeah, when we were talking about this, we were writing notes for this episode, I hadn't appreciated Kyle's point. And maybe it sounds nuanced because of, what you're describing, but I think you're absolutely right that, investing in clinical infrastructure helps us to do both clinical research, right, which tells us a lot about FA at a baseline, but we can then use that information to inform clinical trials, right? So the infrastructure allows us to be trial ready, and now we kind of are able to optimize, right, how we do these trials because of what we learned, right, in the clinical research enabled by clinical infrastructure. So it is
and then what we learned goes back into the beginning too to help inform
drug
further
discovery, drug
approaches.
Exactly.
So, all right.
So we're gonna dive in then to talk about the 2 categories under the grant program that deal with clinical infrastructure, right, and how we're investing in those areas. So the 2 are the natural history and biorepository, so that's one category, and then the second one is outcome measures and biomarkers. So if we start with the first, and piece the 2 apart, right, there are kind of 2 things there, natural history and biorepository. So just briefly for the biorepository, because I think we were talking about this, it makes a lot of sense intuitively, right? This is going to be taking those samples that you guys donate, right, and storing them in a place that can then be used later for research. And so, I actually got a bunch of numbers from Jen that I think really kind of hammered this point as far as how your involvement has really helped to develop this particular research resource for the clinic. So, for example, when we look at all the samples that are collected through these, collaborative clinical research network sites, we have greater than 50 skin biopsies, 800 DNA and RNA samples, 500 serum and plasma samples, 800 cheek swabs, the buccal samples. And in the last 2 years alone, we've been able to share those resources with roughly 12 labs, academic labs, as well as roughly 10 pharmaceutical companies. So, I mean, I think that's really outstanding, right? That's just 2 years alone. Those latter numbers.
You know, and you said more than 800 DNA and RNA samples, and each one of those, each one of those 800 represents a person, right? And so it takes people to donate that stuff to be able to enable this biorepository to enable the research. And so I take great pride in providing my blood samples and skin samples because I know it's being used in this way.
Yeah, and we've talked about how these samples are used in previous episodes, for example, with the skin biopsy, and, Kyle was telling us about the...
Yeah, I learned what a fibroblast is, right? Yeah,
exactly, yeah. So we won't talk about those today, but I just thought it was just awesome, right? The sheer magnitude, like the numbers there, as far as, again, what Kyle's saying, how your contributions really enable research, quite literally, right? So with that, we'll just scoot on to the next category in this bigger category, natural history studies. I think Kyle had some points.
Yeah, so kind of what is the natural history study? Yeah. I think a lot of people out there probably participate in it. I know you do because there's lots of data that comes out of that study, but it's that thing, it's basically one of the main reasons for your annual visit at your neurologist, at your collaborative clinical research network site. We always forget that first CCRN, yeah. So, you go there once a year, and my doctor is Dr. Lynch, so he makes me do the, finger and nose thing, and I put the pegs in the board, and I do the vision test and the walking and all that. And then all that data that comes from me gets put into this database, and it creates a baseline for what FA is and how it affects somebody. And I go back once a year. So we're able to track FA over time and see the true progression in a person. And we're able to see that in hundreds of people because hundreds of people participate in the trial. And so that gives us a pretty darn good idea of how FA progresses over time, so that when we apply a treatment to the people that have FA, then we can tell if it's working or not because we know how it should work without a treatment.
Exactly. Yep.
So so an illustration of how the clinical network enables clinical trials comes from Reata Pharmaceuticals. And I have a quote from Colin Meyer. He is the Chief Medical Officer at Reata. And he says, the investigators and coordinators in the Collaborative Clinical Research Network are incredibly responsive and professional. With their partnership and fair leadership, we were able to identify interested investigators and sites, develop a phase 2-3 protocol, obtain regulatory and ethics committee approvals, and launch a study in approximately 6 months. And that's the true power, I think, of the natural history study and the clinical network, right? That Reata can learn so much from that data and from the network, the experts that we have that have been treating FA.
Yeah, and I think too, this is the full circle, bit that Kyle was talking about earlier, where we have clinical infrastructure in place, i.e. clinical, or collaborative clinical research network sites, and physicians at those sites that can, have generated data and also can lend insight, right, into the baseline, right, our baseline understanding of FA. And that information has then informed, right, the clinical trial design of the MOXIE trial, which I think is pretty amazing, right? That basic research that we were doing, clinical research, that we were doing before, right, is now being used, right, to allow us to have really optimized trials to understand, right, if there's going to be an impact in the FA community with this drug. So, yeah, I think the full circle bit that Kyle's referring to is really, really well exemplified in this.
Well, example. And, one thing we were talking about earlier is if we didn't have a clinical research network, for one, it would have taken a lot longer than 6 months.
Yeah.
And another thing that you mentioned was that Rihanna might not have been interested at all because they're like, we're not going to be able to get this done very quickly, so maybe we shouldn't do it. But the fact that we're able to, that attracts them and more, and more companies. So, let's move on to outcome measures and biomarkers.
So I don't know if you guys remember this from the last episode, but we were using it when, we need to pause and make some definitions here. So when we were making notes for this episode, I totally threw this up because for me, I get a little confused about the difference between an outcome measure and a biomarker. I think as a scientist, I know very well what a biomarker is, but not fully, I didn't understand what an outcome measure is. And Kyle had a really great explanation that I will let him provide.
So an outcome measure is performance-based. I think that's the absolute distinction. A biomarker is something that indicates change in the disease, like something that you would see in the lab, right? And you would track this biomarker to see how an indicator of the disease is progressing. Well, the outcome measure measures how the disease is actually affecting somebody, and it's performance-based. So it's something like the timed 25-foot walk, or the pegboard test, or they ask us all those. They ask us to fill out the surveys. Oh my gosh, and they're actually really useful because they give us an indication of how FA is affecting our daily lives.
And you can imagine it literally too. So outcome measures are used, right, as kind of the benchmarks in a clinical trial for whether or not the therapy is working, right? It's going to measure the outcome of the therapy. And so, Kyle, it's a really great, understanding that you gave me as far as it being performance-based. We want to see that the therapy had a meaningful impact, right? And that's an outcome measure,
So we related this to a metaphor, right? So the little tire pressure indicator on your dashboard, it is a biomarker. It maybe not bio, but it's a marker, right? It tells you that something's wrong with your tire. And if you don't do something about it, you're probably going to have a flat tire.
Right, and that is the outcome, right? The outcome measure is your tire is flat, right? The indicator, right? You can go and look at your tire even when the light is on, and it doesn't look flat, right? But it is an indication that it will eventually become flat. It tells you about the outcome, right?
And like we were saying before, maybe the outcome is the cars that build you to drive 1 mile,
exactly.
Right?
It changes drastically when you have a flat tire.
Right, exactly,
And we had a lot of fun with this analogy, as you can see. But we did want to provide you some examples on the website under this category, right, of biomarkers, just as an example.
So before we go to that, I wanted to talk about surrogate endpoints.
Yes, so before we get to examples, I think that's a really good side point. So Kyle and I were talking about what happens. So outcome measures are used in clinical trials, right, to tell you if a therapy is working. Can you use a biomarker in a clinical trial, right, to tell you if a therapy is working? That was kind of the question that we had.
Right,
I think Kyle got some resolution around that.
So the thing is, if a biomarker can be directly linked to an outcome measure like an 25-foot walk or activity daily living, something like that, if a biomarker can be linked to that, then it's possible that it can be validated as a surrogate endpoint. And that can be, the surrogate endpoint can be used in a clinical trial.
And I think a really good example, just to step away from our tire analogy, but a really good example I think is cholesterol we were talking about in our little meeting. So the idea, right, with cholesterol, there's a lot of data to support that when you have certain changes in your cholesterol level, in particular, increased levels of what's called bad cholesterol, right? That's an indicator of potential poor outcome with regard to your heart health, right? And so you go to your doctor for your annual visit and they check these things regularly, and if they do see, right, if they do see some kind of higher level of this bad cholesterol, that is like a red flag, an indicator that you could potentially be experiencing in future, right, poor heart health. And so there are interventions that they can give to lower those levels, right, to try to, prevent that bad outcome. And so I think that is a really good example of a biomarker. You can't see, right? If you look at me, you can't see my cholesterol levels, right? Not visibly, right? Or if I do any kind of performance test, you can't see it. But it's a biological indicator, right, that potentially, right, I will have poor heart health, right, if it's too high. So that's essentially, I think, a really good example of what a biomarker is and what we talk about when it's a biomarker that we can't see, which I think is a good transition into some of the examples that we wanted to talk about. So, for example, we are funding a couple imaging studies. So one that we wanted to highlight is from Pierre Gilles Henry and Christopher Linglet, and I think it's been going on for about 4 years now. But essentially, they are imaging the different regions in the CNS, right, in your neuronal system.
Central nervous system.
Yes, thank you, central nervous system. But what they're interested in are structural changes that we know are happening with disease progression in FA. And they want to measure very carefully, right, how these changes are occurring over time. It's kind of similar to what Kyle was describing with the natural history study, right? Like, how much change can we expect to see over a certain amount of time? And with that change, what does it correlate to with regard to the outcome measure, right? So this is a biomarker in the sense that you can't see the change happening, right? We know that it's happening. But maybe in future, right, we can use that as a surrogate endpoint, right, in a trial. So if we expect to see this much change, structural change happening in the CNS, right, and we give a therapy to try to prevent that, right, we can observe if it did happen. And if it didn't happen, it might be able to, tell us about the outcome in the end, i.e. if ataxia, right, like we progressed more towards a worsened ataxia. I hope that made sense. There's a lot of technical terms in there.
Yeah, no, and I mean, just thinking about a biomarker, I think the value of a biomarker, even though, like, maybe, okay, a biomarker can't be used as an endpoint in a clinical trial, but in research, when we're testing a drug, when a company is testing a drug, they use biomarkers to indicate whether that drug is doing what they think it should, whether it's making frataxin go up or like changing whatever in the cell,
Yeah, so. I really do think that the tire analogy is pretty great. Like, you can really do think of it as an indicator of what's to come. So you're absolutely right in that in the lab, right, we can do the same, right, as far as using it as an indicator of if our therapy is working as we expect.
Yeah, but and so like the work with Pierre Gilles and Christophe is, a company could potentially use that to give them an indication of how their drug is working based on these images that they're taking of the central nervous system.
Yeah. Yep. If they're interfering with progression of, those changes.
Yep.
Yeah.
So, that is
surprisingly all of our episode.
So
I say surprisingly because we are known for talking a whole lot, but we wanted to save time to have questions, not just for this episode, but also for the program overall. So as you all know, this is, sorry, Jen's handing me a piece of paper here, but as you all know, this is the last episode of our series. And so, we wanted to make sure that we save some space and time to talk about the goal of the program overall. And I think Kyle highlighted it at the beginning very well, so not to go back too much, but essentially we wanted to make sure that the grant program was pretty transparent to you all, given that the money that we raise, right, goes towards funding research. So we wanted you to understand what that meant when we fund research. What are the areas that we are prioritizing? How do we decide on that? And to just parse that out a little bit. But ultimately to end up in where we are right now, right, which is all of this work helping us to enable success in the clinic. And Kyle and I were talking about this idea that when we mean success in the clinic, we mean failing fast. I really like that. Yeah, that phrase.
Yeah, like success sometimes means figuring out that something doesn't work, right? And we need to stop spending time and resources on that because now we know.
Yeah, and we've built the research and clinical infrastructure to do that. And that was kind of the goal of the program, is to share that with you. And so, so we will take questions on both this episode, but then also overall, like what you guys felt about the program, or even ideas about like other series that, you might want to see. So I will stop rambling.
Yeah, so we have some questions and comments and stuff, but while we're doing this, maybe think of what if you were here for multiple episodes, if you watched previous ones, what was your biggest takeaway from this series? And what would you like us to address in the future if we decide to do the skin? So I know I've had a good time.
Yeah.
So
that was a yes. Sorry. All right.
She's like, yeah, I guess.
That's very positive. This guy's great. All right, so just to jump into some of these questions, comments, wanted to give a shout out to Leland Sims. It's the birthday.
Yeah,
really cool. Yeah,
Leland.
Happy birthday. Yeah, and thank you for joining us on Facebook Live. Oh, woohoo! Yeah. Okay, and then we have a question. I think this one's for the episode. So, folks want to know how you can contribute samples to the biorepository. And so I think if you guys go to the curefa.org webpage and you click, I think it's like the research tab, and there's a drop down, you'll see Clinical Research Network as a like listed bullet. So if you just click on that, you can actually see all of the CCRN sites and information, contacts on who to get in touch with to actually go and participate. So that's where I would encourage you to turn to. And we have another one at Natural History Study. So DNA and RNA cheek swabs are collected or special sample collection. I'm not sure that's a question, more so extra information. So we'll get back to that one. All right, Kyle, what do you have?
So I have a question from Alex Fielding. What's up, Alex? Thanks for the question. His question is, do the biorepository samples have a shelf life? And how often do we need to replenish them? So, do biorepository... So, shelf life is less of an issue, but we share and use up samples and do need to replenish the stocks. So, so these samples, so they go to like Merrick's lab, right? And they
Depends on the sample, yeah.
Well, for
the skin biopsies, they will go to Merrick Naparella's lab. So I think we've posted the Facebook Live link that Kyle did visiting the lab.
And he pulls out the thing out of and all this like smoke comes off of it because it's in dry ice.
Yeah.
And I'm like, oh, my cells may be in there somewhere.
Yeah, they are definitely in there somewhere. But those will live for forever in that system. And that's because, that's a skin biopsy, and he's taken the fibroblast out, which can perpetuate for quite some time. So anyways, if you freeze them like that, they will last for a pretty long time. But I think Kyle's point is right, that shelf life is less important because actually those samples get used pretty readily, which is excellent, guys. So, yeah, we have to replenish them quite often, and it's, again, through you. So.
All right. And this question is from Candy Stacks. And we got a handy little answer here from Jen, so I'll read that, and then we'll see what we think about it. But first of all, the question... That's not what I meant. That's not what I meant. I don't know what I meant. All right. Yeah. Here's the question. Can you also discuss the latest natural history for FA children? I believe every 6 months.
Yeah, so I can answer this one. I think you guys saw the announcement, but the FDA gave a special grant to 3 of the CCRN sites, so CHOP, UCLA, and University of Florida, to study FA in children. And so they want to do it on a more, essentially the grant allows them to do it on a more frequent basis, so every 6 months instead of every year. And I think it's going to be very similar to what we've done before, but the idea is that we're going to try to test new measures that might be more sensitive in kids. But again, it will give us greater resolution about the progression of FA in children. So yes, Candy, you are correct. And this will be really good too because it'll inform, just as the natural history study with mostly adult data has informed clinical trials, this will inform future clinical trials in children. So it's actually a really important step in that direction. Yeah.
All right, so we got a question here. I'm looking at the little tablet here, and from Rolf Hill, it seems that biomarkers are more objective and less subjective than, for example, the FARA or SARS score. Let's see, and biomarkers can be used on a wider range of patients. are there enough sufficient biomarkers to assess drug efficacy? And so I think, it's a good point, is that to do the FARS, we have two people, me and Dr. Lynch, right? And so the score is based on his judgment of me, and Dr. Lynch is very good. He's like a machine, man. But there's some obvious variability when you have that, and you have multiple sites, multiple different people doing that. And so I think the point is maybe a biomarker is less subjective, right?
Yeah, and I was thinking too, so the surveys that Kyle was referring to assess how your experience, your daily activities of living, right? Things like that. That's quite subjective, and it can depend on a lot of things, not just the drug that you're experiencing, or that if you're in a trial, right, that you're testing out. And so yes, I would agree that biomarkers are much more objective, and Jen has a really great response that she's telling us to read.
However, biomarkers don't tell us how somebody feels or functions.
Yes, yeah.
And that's the point of an outcome measure, is that's the X factor, right?
Yeah, and I think too, that's why having surrogate endpoints, right? So a biomarker that matches, right, the outcome measure is really important, and we're not quite there yet. And so we've actually invested quite a bit in biomarkers, hence the category, right, outcome measure and biomarkers, so that we can get there. Recognizing that FA is progressive, so again, those outcome measure changes that we see may take a long time, so having some kind of surrogate endpoint to say in a shorter period of time, right, if an intervention is working, because we know that it correlates with the outcome measure, is really important. And we're doing a lot. That's why we have this category, investing in it, to be able to have both the objective and subjective but sound, right, as far as telling us how, if it matches the patient experience, right, having that paired. Yeah.
So I think the point is we need both.
Yes.
Maybe for different and perhaps overlapping reasons, but we do need both.
Yes. And I think...
You might need multiple ones for different...
Oh, I see. So Jen is also emphasizing that, and I think it's actually an excellent point. So depending on your therapeutic intervention, right, you're going to need a different biomarker and or, outcome measure for different parts of the disease, right? So you might need one specifically to understand changes in neuronal function, or one to understand changes in cardiac health, right? Or not more importantly, but another important one is this aspect of diabetes, right? Understanding that intervention as well. So you might need different measures to tell you different things with regard to aspects of the disease.
And part of that point is that there is no such thing as a perfect biomarker that assesses everything, and so I think that the search for the best biomarker for whatever is ongoing, and it's going to be going on for a long time, if not forever, so
I wouldn't say
right. It's
okay,
a
I think we're getting
too.
pretty close. As you can see under the category, we have imaging biomarkers to look at the spine. Over at CHOP in the COE, we have a lot of cardiac MRI markers.
Okay.
So we're
we're constantly perfecting these biomarkers, but we won't need them when we find a cure.
Yes.
So there we go.
There you go. This is getting philosophical, but I like it. I dig it. Yeah. All right. So we have another question actually from Reed and Scott Huckabee. Oh, actually, it's a comment.
No. That's old.
Oh.
Right? That's
the
comment. Read the comments.
Oh, the comments.
So
from Scott Huckabee.
Oh, you want us to read the comments? We are not doing a good job, getting through these comments. But yes, go ahead.
It would be cool to have live episodes that talk about all the fundraisers going on, given month or in the future. How much they raise and how to donate.
Yeah.
By the way, Ride a Taxi SoCal on the beach. February 24th, 2019 is on the calendar, and we're super stoked. Yeah, baby, that's what I'm talking about.
That's a really awesome idea. Yeah, I think it's really cool. I mean, we have it on the webpage, right? And so, but I don't know, I think having it in a collected space and also on social media, like in this format where we're talking about it, I think that's awesome. Yeah, very good idea.
Yep. And we're looking forward to seeing you on the beach again, Scott. It's going to be awesome.
Next year, yes. Okay, and then we have another comment too, Leland Sims.
All right, let's see. Leland, Leland, let's see. Oh, here we go. For future episodes, I'd love to get more specifics about each drug in the pipeline.
Yeah.
I know it's on the website, but having a personal update interests me.
Yeah, I think Jen gives a really good update every year at the patient symposium. But I agree, for those that aren't present at that meeting, yeah, it'd be great to even have that, right, presentation be put somewhere that everybody can see it and there's dialogue about it.
Yeah, because I think Leland's point is like, yeah, this is a pretty graph and there's lots of cool colors, but it doesn't tell the whole story, right?
And it's a lot of text, so you can click on any one of these, right, to like read more about the company and the therapy and understand the science behind it, but there's a lot of bars there, which is great, right, for us, and as far as having things in the clinic, but not so great when you're overwhelmed by the science. So.
And this gets updated, the pipeline gets updated a lot, on a consistent basis, but stuff happens all the time.
Yeah.
And so I think that's a comment that we will take to heart for sure.
Kyle keeps exaggerating, though, the changes that are happening, with the biomarker talk. This is updated, but I would go, it's a really good resource if you want to learn some things. But I agree, breaking it down is important.
We've got the scientists, and then there's the guy who over-exaggerates everything and puts the emotion in everything, right?
Just a little bit.
So maybe we balance each other out well,
Yes, I agree. All right, so let's see. Oh, I think there's one more comment, too. So Alex had a comment about, how to start a fundraiser. Ooh, and we've got a good answer for that one, if I'm reading your comment correctly.
So Alex says, I agree with Leland's specifics into the different approaches. That was, and then, wow, Alex has multiple comments here. He says, it would, some episodes on fundraising, how to get started, event ideas, what's planned for, maybe could be led by some of the families and friends who've experienced different types of successful fundraisers. Yeah. I think that's a really good idea. I like that.
Yeah, I was going to contribute. So I know Anne in the office here really did an awesome job creating this packet about how to, get started with the grassroots fundraiser. And I believe that is on the curefa.org website. So I don't remember where / but
under tools.
Under tools, yes. If you go to curefa.org in the tool tab. But yeah, and it's a really awesome packet to help people get started. But I think a conversation, right, in this kind of format, I think conversations in this format are just really helpful. So I think that's a really good idea. We'll take it under wing. Yeah.
Yep. Awesome. All right.
And I think we actually had one more question. I don't know where it went.
Well, I see one here from Alex, and it relates to the actual content of the episode.
Okay, I think it's the same one.
Oh, yeah, it's written down right there.
Yeah, so Alex asked, can frataxin levels be measured as a biomarker? And the answer is yes. The Center of Excellence for FA at CHOP is working very hard on it. So this is the lab of Dr. Ian Blair. And, he's trying to get really sensitive measures of frataxin levels in different cell types in the blood, so not just blood cells, but like also immune cells, like white cells, things like that, with the goal, right, of this being a biomarker that tells us something about, baseline levels, but then also when we have some kind of therapeutic intervention that's meant to maybe augment frataxin or increase frataxin levels, right, that we can see it reflected in the blood before, again, we see a positive change, right, in an outcome measure. So that would allow for that particular biomarker for frataxin measurement to be a surrogate endpoint, potentially. But we're working really hard on that, and Ian Blair is pioneering that work, so yes.
So, yeah, cool. So frataxin is absolutely a biomarker.
Yeah,
and they're working on it.
Yeah, Kyle and I were talking about this, but, and I'm afraid to even say it. We really like our tire analogy. And I think that one of the other grants that we wanted to highlight, because someone else had a question about this, I don't think it's in this particular episode, but, we have another grant that we're funding of Henry and Linglet. And you'll see it if you look at the outcome measure and biomarkers. And essentially what they're interested in is kind of a step before a biomarker. So we talked about how a biomarker right? It can help us to understand an
indicator,
outcome measure.
yeah.
Yeah, it's an indicator of outcome measure. But there's actually even a step before it. So if we go back to our tire analogy, you can think about, you know, you were driving and you ran over a nail, right? And when you run over the nail, your tire's fine. There's no indicator light, right? But slowly, there's a hole there that allows for air to leak such that then the indicator light turns on. And then you have a flat tire. And then you can't drive as far as you'd like, right? And so, yeah, the nail, right, in the tire is actually something that we can measure. And that's great because that can actually tell us something about the biomarker, right? So the idea is that if we can change the nail not even being on the street, right? If we can somehow, Kyle had a lot of ideas. Giant magnets all over the place, taking up the nails. But the idea is that if you can prevent the nail from putting the hole in the tire to begin with, right, and causing it to deflate, that's also pretty great. So the second grant that we're funding is, you know, their attempt to use imaging, right, to try to understand, you know, what the nail is and whether or not we can, you know, prevent it from being there to begin with. Yeah.
Yeah. I like it.
Yeah, yeah.
Nice.
So,
All
I
right.
think so with that, we are all done. Yeah.
Yeah. So, thanks so much for joining us. This has been really fun.
Yeah.
Just
yeah.
an idea that we had while talking in Angel's office, and then, you know, it kind of turns into this, and it's way awesome to have immediate conversation with people that are watching. I think that's really incredible, and, you know, that's the thing with the FA community is that, you know, we're all in this together. Now, having a discussion like this, I think, is really, really awesome.
Yeah, and I definitely hope that was kind of the goal for me, but that you guys feel empowered, right? That you have a lot of insight now as far as, you know, what we mean when we say that we fund research and how that actually enables us to develop treatments and realize a cure for FA. So I think you guys are all armed now with that information, and hopefully you can share it with others, right? Or when you come across an article or an announcement or something, you can better understand what it means, right, in the context of our treatment pipeline. So that was the goal, and I hope that you guys are all on the same page with us. So
yeah, and also, you know, if you come across something that you don't understand or whatever, you need a little insight, feel free to email us.
You can do Kyle. He's an expert now. Yeah, he totally got this. Yeah.
And then I could go over to Angel.
Yeah, I know. I was gonna say.
You know, we understand this.
Yeah. But yeah, guys, it's been great, and we will talk soon. Yeah, with all these other ideas.
All right, thanks everybody.
Bye.
The finish.
Well, I guess we're gonna stay together because I can't press buttons. Bye,
There
guys.
we go. Bye.